Today we're going to start looking at genetic population changes. So population genetics is a field which quantifies the genetic material of populations. It measures and it counts, and sometimes we will go all the way down to gene sequencing and tracking specific patterns of nucleotides within a population, which requires a whole ton of computing power to do, but we're not doing that in grade 12. Um, we call the genetic information of a population its gene pool. The gene pool is made up of all of the alleles that are in that population. Now, when we talk about a gene pool, just like we'll usually only talk about one particular gene and its alleles at a time, gene pools work the same way. We're not going to make a pedigree that's tracking 5 different genes. We're not going to do a gene pool that tries to track multiple genes unless you're using aforementioned lots of computing power. But we're not doing that. Most of the time when we talk about a gene pool, we're just talking about one single gene and its alleles. So we do have multiple gene pools that operate within a population, one for each gene on the chromosomes. Really important thing to remember about the gene pool is that the gene pool is always twice the size of the population. And population size we give the variable of n. And that is, of course, because each individual in the population has 2 alleles. So if you have 200 individuals, you have 400 alleles. If you have 16 individuals, you have 32 alleles in the gene pool. And we count the 2 alleles that any individual has as being separate within the gene pool because they do not influence each other's inheritance. Inside these gene pools, we have allele frequencies, and this is really the nuts and bolts. Allele frequencies are probabilities between 0 and 1. 0 means that none of the genes in the gene pool are that allele. 1 means all of them, and of course sometimes you have more than 2 different types of alleles. So you could have one at 0.4, one at 0.35, and then one at 0.25. But most of the time when we talk about these population genetics problems, we're dealing with simple Mendelian traits, so we'll just have one dominant and one recessive. And note as well that the gene frequency is not the genotype frequency, and that's why this image on the right-hand side is there. It's actually very important because what we see in the top population is we see that we have a whole bunch of heterozygotes in that population and a whole bunch of homozygous dominant individuals and no homozygous recessives. But in the bottom population, we do have a bunch of homozygous dominant, in fact more than in the top population. We have a single heterozygote, but then we also have 2 homozygous recessive alleles. And the really interesting thing about this one is that these 2 populations, the frequency of the dominant and recessive alleles is the same. So the genotype frequency does not necessarily equal the gene frequency. And one set of gene frequencies in a gene pool can describe multiple very different populations depending on how they're dispersed throughout. But a lot of the time, if we know the gene frequency, we can use that to calculate what the genotype frequency is, at least if it's meeting a special condition we call Hardy-Weinberg equilibrium. We can. And if we know what the allele frequencies are, we can also calculate the genotype frequencies at Hardy-Weinberg equilibrium. So what I have here is I have a very simple Punnett square. Looks like any other Punnett square you normally deal with, but what it's actually showing you is it's showing the frequency of the particular genotypes within that population. We are actually going to get if the dominant allele has a 70% frequency and the recessive allele has a 30% frequency at Hardy-Weinberg equilibrium, which we'll talk more about next video. you'd have 49% of the population being homozygous dominant. You would have 41% of the population being heterozygous, and you'd have 9% of the population being homozygous recessive. And we've just calculated that by multiplying the probabilities together. Now you'll notice those probabilities are between 0 and 1, and I was talking about percentage. Percentage is just 100 times what the probability is. So probability of 0.7 is 70%. And probability of 0.49 is 49%. But it's really important that whenever you do multiplication, you always do it with the probabilities, never the percentages, because you'll get very different numbers if you do.